So there are two broad categories of active transport. The first category is primary active transport. Primary active transport requires the involvement of something called ATP. If you've taken a biology class, you've probably heard of this. So a classical example of active transport is the sodium-potassium exchange pump, which is a protein. So is the thing about proteins, they're so cool. Not only can they form all these shapes, but they can also form all these functional machinery of the cell. So the thing about the sodium potassium exchange pump is kind of like an engine, and its gas is ATP. It uses the energy from ATP to perform its functions. Now, let's rewind a bit. So the thing about your cells is that they have differences in the concentrations of ions inside the cell versus the concentration of ions outside of the cell, and vice versa. So in a typical cell, what you'll find is more sodium concentrated in the extracellular fluid surrounding a cell than there is in the cytoplasm. Whereas in terms of potassium, you actually find more potassium at a higher concentration inside the cytoplasm than in the extracellular fluid. It's not like this night and day, but I'm just showing it for simplicity's sake. Okay, so the thing is that how do you get that imbalance? How do you get more sodium on the outside and more potassium on the inside? Well, the thing about that is like, can you use an ion channel? Well, remember, ion channels help with facilitated diffusion. So if you open an ion channel and have no additional energy, which way are the ions going to travel? And remember, this is facilitated diffusion, no energy added. And which way does diffusion move? From high concentration to low concentration, or low concentration to high concentration? So remember my general rule, high to low, right? So it's going to move from that high sodium concentration. It has an easy, if you open an ion channel for sodium, sodium has an easy time flowing into the cell, no additional energy required. Now, can sodium travel through that same ion channel back towards the outside? Well, remember in my previous example, you have a high concentration here. So when you already have too much of a substance like sodium in the surroundings, it gets harder and harder to add more sodium to the outside. So that just by itself, without any additional energy, that sodium can't join all the sodium that's already there in excess in the extracellular fluid. So it's going to pretty much shove it back into the cytoplasm. So in order to add more sodium to the outside, you need to add energy. Remember, going from high to low doesn't really require energy, but to go from low to high, you need energy. So ion channels cannot make this difference between the sodium concentration in the extracellular fluid and the sodium concentration in the cytoplasm. Now let's look at potassium. Remember, in a cell at rest, that potassium is found at a greater concentration inside of the cell than in the extracellular fluid. So can you use an ion channel to cause this difference? Well, remember, if you want to, this is your end goal. If you want more potassium inside the cell, if you open up an ion channel, all that potassium is going to, again, if just without any additional energy, flow from high to low concentration. So it's going to, all these potassium ions are going to eventually flood out until the concentrations are equal between the cytoplasm and the extracellular fluid. And again, can that potassium travel back into the cell? Well, remember, if there's already a lot of potassium in here, it's going to be harder and harder for additional potassium ions to join the abundance of potassium ions already there. So that would require energy, but remember I said those potassium or those ion channels, they're facilitated diffusion, they're passive transport, they do not provide additional energy. So what I'm getting at here, these ion channels cannot cause that imbalance between and difference between the greater sodium concentrations in the extracellular fluid and the greater potassium concentrations inside of the cell. So this is where the sodium-potassium exchange pump comes into play. And remember, I said it's like a protein engine that uses ATP as its fuel. But what is ATP? So ATP is adenosine triphosphate. So adenosine is a nucleotide, just like in our DNA and RNA section. But here we have phosphate, and again, similar to the phosphate groups found in phospholipids. But it's triphosphate, so there are actually three phosphates linked together. And the funny thing about this phosphates, 1, 2, 3, is that they can actually store energy within these bonds that link these three phosphates. And the bond that links the second and third phosphate, it actually has very, very high energy. So the thing about your cells and the proteins and enzymes inside your cells, they can actually break this bond to release energy that helps it to perform their functions. Now, with active transport, again, we're actually in a cell at rest. You have more sodium on the outside and more potassium on the inside. So how do we get that? Well, I'm going to put that in the background just so that you see that, again, there is typically a lot of sodium in the extracellular fluid and a lot of potassium already in the cytoplasm. So how do we get these sodium ions to join all the abundance of extracellular fluid sodium ions out here? And how do we get these potassium ions to join all these potassium ions already into the cell? This is where the sodium-potassium exchange pump comes in handy. So what do you notice? Notice that it's facing toward the inside of the cell. So what the sodium-potassium exchange pump, well, exchanges sodium and potassium, but let's break it down a little more. So it takes 3 sodium ions, and then it uses the energy from breaking those bonds in ATP. So that provides energy to actually cause that protein, that sodium-potassium exchange pump, to change shape. So now it's facing the outside. So now it can release these sodium ions into the surroundings. And it can actually, so it doesn't stop there. What actually happens next is that instead of sodium ions, you have 2 potassium ions binding to the pump. And it allows, so this phosphate group is removed. And then it changes shape back to its original shape. And now it can release the potassium toward the inside. So if you keep on using ATP over and over again, and repeating the cycle, what eventually happens is that, again, now you notice that sodium is building up on the outside and potassium is being brought into inside of the cells. This is how you have that concentration gradient. So it's due to the sodium-potassium exchange pump and using ATP to add sodium where there's already a lot of sodium and add potassium where there's already a lot of potassium. So it's all the energy from ATP that allows these ions to accumulate where there's already a big presence of these ions.